Welcome to our lecture presentation on the reproductive system. And this is our final presentation in this section here. So let's get started. So let's look at the male reproductive system first. And we're making sperm cells here. So sperm cells, they're made in the testes here. And sperm cells are actually manufactured in the testes here. And then they're transported through a number of tubules here. So I have the epididymis here. Epididymis connects to the vas deferens. Vas deferens comes out here, passes through the prostate gland, goes through the urethra, and with ejaculation now leaves the body here. And I've got a couple other glands that we need to be aware of here. Here, seminal vesicles make some fluid content of the ejaculate here. I've got the bulbourethral gland. And what the bulbourethral gland, remember this, but this one's an important one. It secretes a fluid that neutralizes the pH of the urethra. Because remember, one of the jobs of the kidney is to get rid of all the extra protons you have in your body, mix this in an acidic environment that can damage sperm cells that you're trying to pass through here. No worries. The bulbourethral gland neutralizes that pH. So when the sperm come down here, they pass through unharmed here. And now let's go on to our other gland. Here's the prostate gland. The prostate gland actually makes much of the fluid in the ejaculate. It's actually a fluid, this white milky fluid secreted by the prostate gland because although in the ejaculate you're making hundreds of millions of cells in there, sperm cells in there. Most of that fluid really comes from the prostate gland. It is this fluid absolutely essential here. Seminal vesicles here. This secretes the fluid, and this nourishes the sperm. Gives the sperm a little bit of energy before it goes off on its trip there to find the egg here. So here's a close-up of the testes here. I've got a number of these tubules here. So I start off here with the seminiferous tubules. And the seminiferous tubules, these are going to be where the sperm cells are manufactured. There's stem cells here. They're continuously making new sperm cells throughout the life, of the male here. And here's the site of manufacture here. When these sperm cells are made, they're passed here, the epididymis. Ready testis goes to the epididymis. Epididymis goes up to the vas deferens and goes up here, up the spermiana cord here, and now, on its way, as we saw in the previous picture here. So let's take a closer look at the seminiferous tubules here. Just, how are these cells manufactured here? So throughout the male's life here, I've got these stem cells, and the stem cells are continually dividing, making more sperm cells. So more sperm cells are born all the time here. And these sperm cells are formed from spermatogonia, and these are located in the seminiferous tubules. Spermatogonia divide and to form primary spermatocytes. Primary spermatocytes then undergo meiotic division. Because remember, one of the jobs of making a germ cell is that, humans, you're going to start off with a diploid chromosome count, what they call 2N chromosome count. In humans, it's going to be 46 chromosomes here. But when I want to fertilize an egg, I need to have 23. I get to reduce the number of chromosomes here because 23 chromosomes come from dad, 23 chromosomes come from mom. I fuse those two nuclei together, I get a total complement of 46 here. So within all of us, in our 46 chromosomes, we've got one chromosome from dad, one chromosome from mom. And these are sort of homologous chromosomes. They're matching chromosomes. They've got the same genes in the same location. One happened to come from mom, one happened to come from dad. In the process of making sperm cells, then males now in this generation, they're going to pick one of those pairs. Doesn't matter whether it's originally from mom, originally from dad. Take one of those, put it in the sperm cell, packages it up. And what we want to understand is the sperm cell. This is a DNA delivery device here. This is its sole purpose here. It is specialized for this task. And it turns out, as we're going to see, this is a really difficult task. The life of a sperm cell, chances that you're going to fertilize an egg are minuscule here. But okay, let's continue on with our tour here. So here's meiosis. So unlike mitosis, which was to make more cells here, meiosis, I'm trying to make a sperm cell with half the number of chromosomes here. I got 23 chromosomes in the case of humans here. So I'm going to start off with my seminiferous tubule. In the seminiferous tubule here, I'm going to have the spermatogonium. Spermatogonium these are stem cells. They're making more of these cells here. And then it undergoes actually 2 divisions in succession here. So the first meiotic division, I take that primary spermatocyte, I divide that up, and now I have half the number of chromosomes. And the DNA has replicated here. So I've got the sister chromatids here. But now instead of pulling these things apart, now I'm going to split them apart. So I get, one complete set of 23 chromosomes here that are replicated. The sister chromatids are there. The other one here, another, 23 chromosomes here with the sister chromatids. The second meiotic division here happens when those sister chromatids are pulled apart. And now I have the N number here, but now I only have 1 chromosome, no sister chromatid here. So each of these are going to have half the number of chromosomes as the original stem cell here. So, okay, this is exactly what I want. Now, these early spermatids, they're going to differentiate. They're going to become sperm cells. They're going to package up their DNA into this little head here. They're going to form a tail here. And now they're attached to the inside of the seminiferous tubules as sperm cells here. Now, this sperm cell is so specialized here that, it really can't do anything except, deliver DNA. The chromosomes, the 23 chromosomes, the haploid chromosomes, are compacted here. They made as small as possible to make this an efficient transport device here. And so you can't read any genes here because everything's packed too tightly here. I've got a little sleeve here of mitochondria to power the tail, so I can swim around here, swim across, the expanse here to find the egg here. But it's a limited supply here. So as this meiosis happens within the seminiferous tubules, I'm coming down here, meiosis 1, meiosis 2, I'm maturing to the sperm cell, and they're attached inside the seminiferous tubule here. But at that point, the tails aren't turned on yet. The tails are immobile here because, you don't want to waste the battery, because if I waste the battery here, it could be a long time before, these guys are called upon to, to swim up, swim upstream to find the egg here. Don't waste the battery. The batteries are turned off. There are other cues along the way, just before ejaculation, that's going to turn on the batteries here. These guys start to swim, and okay, now sperm can be on their way with a full complement of battery charge here. So the problem with the sperm cell here is, we talked about how difficult a task a sperm cell has. And one of the things that's going to make it really difficult for the sperm cell is that, the sperm cell is made by dad here. And, it's got, dad's genome where this is coming from. But when the sperm cell enters, the female here, then it can't be wearing dad's hometown uniform. It's recognition site. Because if it did, mom's immune system would say, this is not the hometown uniform. It'll just automatically destroy it. No sperm would ever survive here. So when the sperm are manufactured in dad, they are manufactured without their recognition molecules here. They are manufactured without the hometown uniform. So that's good. When the sperm cell get the mom, no hometown uniform, then they're still going to be susceptible to destruction by mom's immune system, but not as much because they're not triggering the full response because they don't have a foreign antigen on the surface here. Okay, well, that solves one problem, but the other problem is when the sperm cells are made in dad, then it's not wearing a hometown uniform, so dad's immune system could destroy this before they even, complete the maturation process here. So the solution to that is that when the sperm cells are manufactured, they are manufactured in this matrix here. This is called the blood-testis barrier here. So I've got this barrier is made out of these cells that are called sustentacular cells. So the sustentacular cells are here, and here are the germ cells for the sperm production. So here's spermatogonium. Here are the spermatotids coming up, and they're manufactured into sperm cells here. all of this manufacturing process of the sperm cells in the male, in dad here, is done within this matrix. So they never come into contact with antibodies from dad because they're isolated here. And they're just going to move their way through this matrix of sustentacular cells. And here they are, attached in the lumen of the seminiferous tube. As they park there, everything's assembled, the tail is ready to go, the batteries aren't turned on, so they're just in a state of waiting. And now they're going to be transported through that tubular system, moved up into the body, and now ready for the ejaculate, and I can send those guys on their way, and now everything's fine here. So remember, the blood-testis barrier, very important because it protects the sperm cell from dad's own immune system. It enables the sperm cell to be manufactured without a hometown uniform, without the antigens here that would, trigger mom's immune system. So, okay, I've made this accommodation here, and so now sperm cells are ready to go on their way. So here's my sperm cell. Here's the head. I've compacted that DNA down to the smallest possible, pellet that I can to get the least resistance as these guys, try to swim toward the egg here. I've got here the mid piece. This is where the mitochondria are. They're all compacted here. This is where the DNA is going to come from. A limited supply of energy, so I've got to make sure I don't run down the batteries before it's time to make the swim, to find the egg here. Here's going to be the tail. It's going to be filled with, microtubules here that can now make movements that's going to propel this cell forward in order to find the egg here. At the top of this, there's going to be this little structure called the acrosome. And what the acrosome is it contains a bunch of enzymes. Now, when the sperm gets to the egg, it's not like, the door's going to open and the sperm's going to just, swim right in there. The egg is protected by a barrier here, and the sperm has to dissolve their way into that barrier. And so the sperm has to arrive there, and the acrosome, when it contacts that barrier, it breaks open, and the digestive enzyme comes up and starts to eat a hole within that within that protective barrier. When that hole is big enough, because enough sperm cells have released enough of that digestive enzyme here, now a sperm cell, and one and only one sperm cell, is going to be successful in penetrating the egg, contributing its now haploid complement of DNA into the egg. And as soon as it does that, a second barrier comes out, prevents all the other sperm from coming in here. Because in order to be successful, I've got to have a union of only one sperm and only one egg. If I've got an extra sperm cell in there, then that's going to be lethal. That fertilized egg, which we call the zygote, is not going to survive, and that's going to, it's going to perish. It's not going to work here. Okay, so let's look at some terms we have to know here. So we got to know this one, oogenesis, and this is the process of egg formation in producing eggs that are haploid. Half the number of the normal chromosome count. So diploid in humans is 46. Haploid in humans for the egg and for the sperm, this is going to be, oogenesis, process of making the egg here. Spermatogenesis, this is the process of making the sperm. And again, sperms have got to be haploids. I put two haploids together. I got a diploid zygote. And the zygote is a single cell. And so at the fertilization, you consist of exactly one cell here. And now that zygote is going to start to divide and divide and divide. And from that single fertilized cell, this is where the offspring is going to be generated with the trillions of cells that make up that offspring here. And so it's all got to start with one sperm, one egg coming together, forming the zygote here. The zygote is going to mature and into the offspring individual here. Let's look at another term, ovulation. As the egg is being produced here, it is undergoing meiosis 1, undergoes meiosis 2, and when it's ready, now this oocyte, this egg ready to be fertilized, is going to be released from the ovary here, and it's going to enter the uterine tube, awaiting fertilization by the arriving sperm cell here. Fimbriae, these are like finger-like projections on the uterine tube that are going to, suck up this oocyte, make sure it's going in the right direction, put it into the tube, and start it moving towards the uterus here. So the oocyte, it's got no propulsion of its own. The sperm does, but the oocyte does not. The oocyte, that must rely strictly on, ciliary motion by these cilia located on the fimbriae, which are the finger-like extensions of the uterine tube. Vulva, female external genitalia, external genital organ. Vagina, muscular tube that extends from the uterus to the outside of the body. I got two labia, labia majora, the larger folds of the female reproductive organ on the outside. Tucked away inside a bit is labia minora, internal labia minora here to the external majora here. Clitoris, a projection found on the anterior end of the vulva here. And the clitoris, this is actually, during embryology, the cell that gives rise to the clitoris, if you're female, higher concentration of estrogen, then clitoris is going to form, works great for sexual stimulation, everything's going great. If you're a male, higher concentration of testosterone, this exact same piece of tissue then develops into the penis here. So when you look at all of the pieces of male and female reproductive, there's nothing new. They've just been retasked, been reshaped here. The labia here, these in females, this is the labia majora, the labia minora. In the males, these are going to differentiate into the scrotum here. So we're still dealing with the same box of parts here that we've always had. We've just changed the developmental pathway. More estrogen, I'm going to become female. More testosterone, I'm going to become male here. Vestibular glands, your secretory glands, and in females, and these correspond to the bulbourethral glands in males here. So again, it has a different name, but embryologically it comes from the same primordial tissue here, and secrete fluids here to help the female reproductive process here. So let's stick with our male here. So what's going to start things? The maturation of sperm cells is controlled by hormone molecules here. So the whole process, it doesn't start in the testes, it starts up here in the hypothalamus here. So in the hypothalamus releases GnRH, gonadotropin releasing factor. And gonadotropin releasing factor goes down to the anterior pituitary. And in the anterior pituitary, it's going to cause those cells to release two hormones into the circulation. FSH, follicle stimulating hormone, and LH, luteinizing hormone. And they're both essential to sperm production here. And let's think about this here. LH is going to go to these interstitial cells and cause those interstitial cells to release testosterone. And testosterone goes down to the testes and says, okay, make more sperm cells, get that meiosis going here. We need more sperm cells here. FSH comes down here, it goes to the testes, and it collaborates with testosterone. It says, okay, two signals that say work together to produce, the large number of sperm cells that are needed in the reproductive process here. Now, think about this. It says follicle stimulating hormone and luteinizing hormone. So males don't have follicles. LH, we don't secrete milk. So why they call it follicle stimulating and hormone and luteinizing hormone. And the reason is that embryology really started off studying female, organisms here. And they found this stimulated follicle. It was named FHH. They found LH. It stimulated milk production. So, okay, they called it LH. Then when they got around to studying males, they got the absolute identical molecule, FSH and LH. So it made no sense to give them a different name or to rename the original molecule. So the name stuck here. So although it's FSH and LH, it has its function in the male here. And this is the theme we're going to see throughout. We've seen this throughout our study of physiology. It is that nature doesn't really reinvent it. It can retask them. So in the female, this stimulates follicles. In the male, this stimulates sperm production. Same exact molecule. There's absolutely no difference. Just what are the receptors, and what are the receptors telling these cells to do here in the male? Make more sperm cells here. Now, there's a negative feedback mechanism. So both testosterone and the second molecule here, and the second molecule is going to be called inhibin. The inhibin here is going to be released by these interstitial cells. They go up here and they're going to go up to the production site. Anterior pituitary says, okay, enough with the FSH and the LH already. We've got enough sperm cells here. puts a halt to that, goes up to the hypothalamus, enough with the GnRH here. We've got enough sperm cells here, and this is going to regulate. Make sure I got just the right amount here, because too much testosterone is, that's going to be detrimental to the organism. It's just like anything else on the body. Too little is no good, too much is no good. I gotta get just right. I rely on negative feedback to set that level, to get the exact right level, to get the exact right level of sperm production. And now we're on our way to having, productive or functional sperm cells ready for delivery, in the ejaculate here. So what are the jobs of testosterone here? So, generates male hair pattern and distribution. There are different hair patterns in males and females. This is because you've got testosterone here. Lowers the pitch of the male voice, thickens the skin, generates male pattern and extent of muscle development here. So, the testosterone, higher testosterone male just makes for, more muscle development, and that's just a function of this hormone here. Generates thicker and more robust bone growth. So all of these things. Now, it's important to realize that both males and females have testosterone. You need testosterone. And both males and females have testosterone receptors. And so what would happen, here's the testosterone, high testosterone male driving these events here. What would happen if you injected a very high dose of testosterone into a female here? Then what would happen is that I have to go to very high levels, because females have testosterone receptors, just not as many as they are in the males. I have to go quite high here. And when I do, the females start to exhibit all of these traits here, and it's just, hormone can drive things one way. Now, the problem, the problem with that is the hormone molecules, both testosterone or estrogen, these are very powerful molecules here. So, injecting very large doses of testosterone into a female, is not without consequences. You better have a really, really good reason to do that, because, there is a very high risk of, incurring a number of disease states and pathologies from having testosterone or estrogen levels too high here. So, but just, as a statement that both males and females require both testosterone and estrogen. It so happens males secrete more testosterone, males have more testosterone receptors, females secrete more estrogen and have more estrogen receptors. But the basic machinery, the ability to respond, is identical in males and females here. So from a physiological standpoint, males and females aren't that far apart. From our day-to-day, we make a big distinction whether somebody's male or female, but physiologically, not so much, So let's look at the female reproductive system. And here, the ovaries are here. This is where I'm going to make the oocyte here. And, the ovary, and like we said, is assembled from the same box apart here. So in development, the cells that would in the male become the testes, they have become the ovary. More estrogen made these guys form an ovary instead of a testes. And so here, I've got the uterine tube here. Here's going to be the uterus, vagina. I can see, the labia here. And here's the clitoris, right at the, anterior end of the vulva here. So the parts are here. And like we said, the ovary, that came from the same tissue that made male testes here. The clitoris, that came from the same primordial tissue that made the penis here. The labia, that came from the same primordial tissue that made the scrotum. So everything, has its corresponding part here. And now we got a female reproductive system. Now, here's going to be the uterus here. This is where the egg is going to mature into a embryo and a fetus here. And this opening from the uterus to the vagina, then this is going to be called the cervix here. And cervix here is the entry point here. So if I draw a circle around this whole area and say, name the structure, the answer is going to be cervix here. All right. In the female, we still have meiosis. I'm still trying to make a haploid, ovum, haploid oocyte here. But although the process is similar, the way it goes about it differs. So remember how in the male, like I had the stem cells that were continuously making spermatogonia, making more and more of these primary spermatocytes here. In the female, all this action happens before birth. So during embryonic development in the female here, I start off with an oogonium. I make the primary oocyte, and I have these primary oocytes. All the primary oocytes you're ever going to have in the female are present at birth here. You know, you do not have the capability of making any more here. In childhood, this is dormant, nothing happens here. But then when you come to puberty here, then what happens is these primary oocytes, they start to be moved into the meiotic division cycle here. So here's my first meiotic division, just like we saw in the male. But now there's a difference. Because remember in the male, during meiosis 1, I had 2 successful daughter cells here. The problem with the female making the oocyte here is that there's a lot of machinery, a lot of enzymes, a lot of reactions that have to go on in the oocyte here, in order to be a successful recipient of that sperm cell. And there's just not enough to go around to form 2 successful daughter cells here. So one of these guys becomes the secondary oocyte here. It's now still in an arrested state. And the other one becomes a polar body. And this polar body is incapable of being fertilized. That part of the genome has been completely turned off. This is just going to die and it's going to be discarded. You're just not enough resources. I only got 1 here. And now here I go into meiosis 2. And again, in that second meiotic division, again, only enough resources to make 1 ovum here. The other one becomes the second polar body that is discarded and reabsorbed by the body here. So the question is, well, if you have all of the primary oocytes, when you're born and you can't make any more, then what's the how close what's the probability that a female is going to run out of oocytes sometime in their life? And the answer is that when you run the numbers, there are hundreds and hundreds of thousands of these primary oocytes, and ovulating once a month for your entire lifetime, you're still going to have hundreds of thousands of these primary oocytes left. So you don't stop ovulating because you've run out of primary oocytes. You stop for other hormonal reasons, not because you've run out of these cells here. And got plenty to go around here, so no worries there here. So here, primary follicle present at birth, and primary follicle is going to be selected to become a growing follicle, a mature Graafian follicle, and finally, release the oocyte into the fallopian tube here. Now, there's going to be a selection process. There are these hundreds and hundreds of thousands of primary follicles here. Then, basically, 1, maybe 2 of these follicles are going to be selected, and they're going to go through these developmental process here. As soon as these guys start to develop, they're going to be releasing inhibitory molecule to go see all of the other follicles and say, I got this. I'm on my way. Do not become a growing follicle. And it inhibits everybody else. Sometimes 2 of these guys get through. And now in humans, and now I've got 2 of these coming through here. And so that's going to be what you call dizygotic twins. I can have sperm cells fertilizing 2 different, oocytes here. And these are the non-identical twins because they came from separate oocytes that happen to be implanted at the same time here. Here's the oocyte itself here. And so here are my primary follicles. I'm going to mature into the follicle cells. 1 or 2 of these, let's just say one of these guys gets selected. It's going to start to go through the developmental process. And now it's going to be cycling here, responding to levels of estrogen and progesterone here. It makes the Graafian follicle. And now there's going to be a separate cue. And that separate cue happens to be there's going to be a peak in those 2 hormones, LH and FSH. And when they get those cues, the oocyte is released. Here's going to be the fallopian tube, the uterine tube. These are the fimbria we talked about. There are all these cilia here. The cilia create a current, suck the egg up into the tube, send it on its way down the tube here, looking to be fertilized by the sperm cells. And if it's fertilized, it's going to successfully implant within the uterus. And now pregnancy is underway here. These cells that are surrounding the oocyte here, they persist for a while. And now this body is going to be called the corpus luteum. And this corpus luteum, it's persisting here, and its job is going to be to secrete more progesterone and estrogen. Because as soon as the oocyte is ovulated here, enters the fallopian tube, this thing starts to make progesterone and estrogen. And what it's telling the body, it's sending those hormones to the uterus and says, get ready because the oocyte is on the way. Become thicker, become prepared, develop your vasculature. Get ready to implant this oocyte here because it's on the way here. If that egg implants, then the uterus itself takes up the job of making progesterone and estrogen, and now pregnancy is on its way. If the egg does not implant, then corpus luteum only persists for 3 or 4 days. It then degrades and goes back to be recycled, becomes the corpora albicans. Now, no more progesterone and estrogen from the corpus luteum. Cycle starts all over again, and now primary follicles start to be advanced here, and we start the cycle again here. All right. So here's the long view of this. Here are my ovaries here. Here the cycling we just looked at, ovulation, and look at this. The ovulation, releases the egg basically into the body cavity here. And now this part of the uterine tube, of the fallopian tube, it has this cilia. It basically sucks up that egg and moves it into the uterine tube here and sends it on. It keeps a current going to send it on its way here. But if you think about it, and when you look at what is the ovulation process actually like, it's pretty brutal here. This primary oocyte, or the secondary, this primary oocyte is ovulated up here, and it just blows itself right up to the wall of the ovary. If you look at the ovary of an adult female here, what you'll see is these little scar tissues all around the ovary here, where this ovulation has taken place here, and that's a perfectly normal, perfectly harmless situation here, and this is how it goes here. But then the other thing is, it's released right into the body cavity. If these cilia don't work, then these guys have no means of propulsion. They are just going to float around in the body cavity. It's going to be completely non-functional here. It also means that, look, I've got a continuous passageway. I can go down here, go down here. I can go through the uterus here. I can go out the vagina. I've got a completely open port from the opening of the vagina coming all the way up here, leading all the way to the inside of the body cavity. And does that mean that pathogens can come in here, crawl all the way up here, get transported up here, and end up in the body cavity and produce some very serious lethal infection? The answer is yes, it can absolutely do that. So how's that going to be? Why don't all the females die from, septic infections of the body? Well, here is the uterus. Here's the cervix here. And right at the cervix, there's going to be a mucus plug here that's made here. And so there's a physical barrier. Anything trying to crawl up here gets into that physical barrier, gets tangled up here. In that mucus plug, I've got all kinds of antimicrobial, compounds in there to destroy anything that's coming in. So I am protected here, but nonetheless, it is a vulnerability here that, a pathogen can make it all the way up to the digestive system and enter the, body cavity and, produce a lethal condition here. Here, I've got some of the ligaments that are holding everything in place. I hold the ovary in place with an ovarian ligament. I hold, this broad ligament holds the uterus. So everything's being held in quite precise locations here, so that when ovulation happens, fimbria is right there. I can suck this thing up here. Now, the part we have to understand is not, necessarily expected. Yeah, I've ovulated. The egg is coming out here, and now the oocyte is coming down here. The oocyte is going to be fertilized within the fallopian tube. It's not fertilized in the uterus. It's fertilized here, and it's going to take it another 2 to 3 days before it makes it into the uterus. And now, if it's been fertilized, it can implant into the uterus. And this is where the embryo will develop, where the fetus will develop, and now, pregnancy is going to be on its way. And here's my hormone complement here. So here's the thickness of the uterus here. So here is your period. Here's menses here. Starts at day 1, lasts 3 to 5 days here. And what's going to cause that uterus to build up its lining and make it thicker and thicker, get ready for the egg so it can implant here, build up this tissue here, bring in lots of blood vessels here, prepare the way here for the implantation of the oocyte, is going to be this increasing concentration of estrogen and progesterone. So the more estrogen, the more progesterone that happens here, the thicker this lining becomes here. I can also see here, this is the other hormones we talked about, FSH and LH. And so things are coming, and I get this spike of FSH and LH here, and this spike of FSH and LH is going to be what causes ovulation to occur. So in experimental animals, if you block this spike of LH and FSH, ovulation does not occur, and the oocyte stays stuck inside of the ovary here. And that is actually a pathology. That's something bad here. So you want this thing, as soon as these guys develop, give the spike, have the oocyte come up, sucked up by the fimbriae. Here's going to be my corpus luteum being made here, making more estrogen, making more progesterone. Here's the thickness of the uterine wall getting thicker and thicker and thicker. And now this enables this structure to deliver more energy, to the developing fetus here. To enable it to take away, waste products more efficiently. And this is what's going to happen here. So if the egg implants here, then this uterus starts to make its own estrogen progesterone. And these numbers keep going up here, and pregnancy is underway here. If the egg does not implant, it's not been fertilized here, so there's no signal to implant, it will not implant. If it does not implant, then estrogen and progesterone levels start to fall back down. When they fall back down, now here's menses, here's you having your period here, and cycle starts all over again, and we go back through here. So in humans, this typically takes something like 28 days. If your period is not exactly 28 days, that's not a pathology. This it just varies between people here, and it's even in an individual, it's not necessarily precisely every 28 days, but in a general cycle, this is going to be what we see. And so we want to remember, this when these LH and FSH peak occurs, what it causes ovulation here. We want to remember what estrogen and progesterone increasing levels cause. Increase in the thickness of the endometrial lining, getting ready to implant that egg here. As soon as that comes down here, endometrial lining degrades away here, and I start my cycle all over again here. Here's the union of the sperm and the egg here. So here's the sperm coming up on the egg, and the egg got this protective barrier here because this is still floating out in the environment here. It wants to protect itself against pathogens. It wants to protect itself against anything that might cause it harm here. The sperm have to come through it, and now the sperm have to have that enzyme that's going to allow it to dissolve its way through this protective barrier. As we said, that's contained in the acrosome here, and when the sperm comes into contact with this barrier, the acrosome breaks down, and now it's going to release that digestive enzyme here, and it dissolves its way through this protective barrier. And it takes several hundred of these guys to release enough enzyme to make a channel through this protective barrier, and one and only one, the first guy that gets there, as soon as it penetrates that egg membrane here, it's going to release its DNA. Its DNA is going to go into the egg here, and at that time, a secondary barrier is erected, keep everybody else out here, and only one sperm can be allowed to enter the egg if this egg is going to be able to survive here. So let's say I got, the gang here has released the hormone. I've dissolved it through here. One guy makes it through. DNA gets in here. Then this point at which the sperm DNA has entered the egg, to join with the egg's, haploid complement of DNA. Now we're going to have a diploid zygote here. Now the structure is called the zygote. And at that point, the zygote starts to divide by mitosis. It starts to make more cells here. I've got one cell. I've got to make trillions of cells to make an offspring individual, to make a fetus here. And now that process of cell division is on its way. So let's back up a step here. Here's the oocyte being released, it's being ovulated here. The cilia on the fimbriae have sucked this thing up, created a current. this thing's coming up here. And here is where the egg is going to be fertilized, in the fallopian tube here. So the sperm have to swim, all the way up through the uterus here. It's got to make it all the way through the fimbria. Remember, the sperm is swimming against the current here. So it's got to swim against the current here, and it's going to fertilize the egg here. And now I've got the zygote here. So here, fertilization has occurred, and these guys start to divide by mitosis. I get the first cleavage here, and so I used to have one zygote. First cleavage happens in the first day, and now I got one cell became two cells. And the second division here, second day, two cells become four cells. Four cells, they all divide, four cells become eight. And what's amazing when you watch this process, you can watch this on a laboratory slide here, is that going from 2 to 4, both of these cells divided exactly the same time. Exactly the same time, 2 become 4. And the 4, each one of these 4 cells, they responding to a cue, they divide at exactly the same time, they become 8, and 8 becomes 16. And on the 32 cell stage, that breaks down, everybody then becomes on their own timing schedule here. But they're still dividing and dividing and dividing away. And pretty soon, I've got this solid ball of cells here. And the solid ball of cells, this is going to be called a morula. And the morula, because the people that looked at this first, they looked at this solid ball of cells, and it kind of looked like a raspberry, because it had all these things on the outside. So that's another name for that particular configuration. So it's called a morula here. Okay, so I got the solid ball called the morula here, and now development is on its way, and it keeps on developing here. So the morula eventually is going to change from a solid ball of cells to a hollow ball of cells. The cells are going to migrate all by themselves, and they're going to pool themselves on one end here. It's a hollow ball here, it's fluid filled, and now this is going to be the blastocyst. And here, 3-4 days after fertilization here, this blastocyst is going to be what's going to be enabled to adhere to the uterine wall. And the job of the uterine wall is going to be to nourish the development of this blastocyst. And now, I'm not a single cell. I'm hundreds of, I got maybe 100 cells in here. And here, this clump of cells here, this is called the inner cell mass. It's also called embryonic stem cells. So this is what the embryonic stem cells are. It's this little pile of 100 or 200 cells here that are present at the time of implantation of this blastocyst into the uterine wall here. So here we are, inner cell mass. Here's going to be my blastocyst, and it's surrounded by these other cells called the trophoblast here. And it's sticky. It's only going to be sticky here if it's been fertilized. If it's not been fertilized, then the oocyte just goes through here, cycle starts all over again, resets fine. Here, because it's been fertilized, now it's going to stick to the uterine wall here. And now these cells, these trophoblasts, they start to differentiate. They start to suck this egg actually into the uterine wall here. So the trophoblasts, what the job here, they have to form a barrier around the egg here. Because remember, this is a zygote. This has got DNA. Half its DNA is from mom. That's fine. But half its DNA from dad, meaning that all of these cells, they're not wearing mom's hometown uniform here. So if mom's immune system were to see this zygote here, it would destroy it, because it says they do not have the hometown uniform. Destroy this, and pregnancy would never occur here. So here, the trophoblasts, they're making a barrier that separates the developing, fetus from the rest of the system here. And now, as I bring this in here, now I get the development of the blastocyst. It becomes an embryo, becomes a fetus, and now in 12 weeks, now it's kind of recognizable as a human here. I've got fingers and toes, and the cycle here. So I've taken haploid from dad, haploid from mom, formed the zygote. Zygote is diploid. The diploids are divide, and it's an amazing task. From one cell that's identical here, I've got to make an organism here with trillions of cells here, and they have to have different functions. They've got to be placed in the right place. And so let's look at how this developmental mechanism makes this all possible here. Now, as soon as that blastocyst embeds itself into the uterine wall here, then the uterus makes this hormone, human chorionic gonadotropin, and it makes a lot of it here. So human chorionic gonadotropin is coming. Here's estrogen and progesterone being made by the uterus, so that's coming up here. But human chorionic gonadotropin, this HCG stuff, this is only really made in these quantities, at the initiation of pregnancy here. And so what that means is that, okay, it's a nice clean signal here. So when you look at those pregnancy indicators that you can just buy at the store there, what they're assaying for is the presence of human chorionic gonadotropin. Because if you see a big spike, and it's a big spike, so it's easy to detect here. If you see a big spike of that, it means, okay, pregnancy is on the way. You don't want to make a pregnancy test out of estrogen and progesterone because these things are going up and down all the time. You could get a lot of false positives here. But human chorionic gonadotropin, as soon as I see that, I'm pretty sure, okay, pregnancy is on the way here. And what the job of HCG, human chorionic gonadotropin, is, it's going to inhibit the pituitary from secreting LH and FSH. So it says, okay, enough with this cycling already. Pregnancy is on the way. Do not, ovulate anymore. So I can inhibit these things here. Estrogen, progesterone, their levels are coming high. They also are feeding back into the system and says, do not, release, do not initiate any more maturation of these, Morgonia here, because pregnancy, so it really puts a stop to the cycling, the normal cycling, reproductive cycling of the cells within the ovary here. And so that's why, birth control pills, they're deliver low doses of estrogen and progesterone here. Because when I deliver these, it goes back into the system, goes back to the hypothalamus and the pituitary. So do not release any of these molecules here that are going to, initiate, ovulation here, because pregnancy is on the way here, and pregnancy is ongoing here. And so that's, how those birth control mechanisms work here. I've got my single cell zygote, so it's dividing mitotically. So I'm making more and more cells here. Here's my, embryonic stem cells here. And let's look at the embryonic stem cells. And people have done this experiment in mice, and they've done it in birds here. So they can see these embryonic stem cells that are being manufactured here. And what happens is that these guys, they wanted to know what is the fate of these cells? What are they going to become here? And so they would go in here, and you can go in here, experimentally, and you can destroy one of these cells here. So let's say in a mouse embryo, they go in here and destroy one of these cells here. They put it back into the mouse. They let the mouse come to term. Mousey is born. Says, what's wrong with the mouse? And absolutely nothing is wrong with the mouse. There's nothing missing behaviorally. Everything, mouse is just perfect here. So they go and destroy 2 or 3 or 4, and still nothing. And you have to destroy quite a number of these cells. There are only maybe a couple hundred cells here. You got to destroy like a couple dozen cells here before something happens. And what happens when you destroy too many of these cells? That now this developing oocyte will not survive, and it dies here. And what that told them was that at this stage here, with these embryonic stem cells here, that these cells are what they call totipotent. They can become anything. They have not received their calling. They have not received their instruction. So any one of these cells could become any one of the cells in the body. It's just a matter of, what are the ligands? What are the cues that are going to tell you? Are you going to become the head? Are you going to become the hand? Are you going to become the nervous system? What are you going to become? Those cues are going to be developed as the system progresses here. So I've got this inner cell mass, embryonic stem cells. And this is why people that were looking for ways to generate cells that I could put into a living person as therapy, you need, missing neurons, or you need missing cardiac muscle, or you need, missing liver cells. Can I take these embryonic stem cells? And if I can figure out what are the cues that I give the cell, can I actually manufacture the missing cells and put them into the person and have them survive? And the answer is, yeah, that can be done here. I mean, it's really hard to do, but people have figured out how to do that. And this is why they were so interested in this embryonic stem cells as a means of treating patients. That before that, there was no other way to treat these patients. And here's a way that they could do that here. Okay, let's get back to normal development here. Here's my inner cell mass, my embryonic stem cell. They continue to develop, and they make this little flat disc of cells here. This doesn't really look like anything yet. It's a flat disc of cell, and this is called the embryonic disc here. And the embryonic disc consists of 3 layers. There's the ectoderm, the mesoderm, and the endoderm. So endoderm, mesoderm, ectoderm here. And these are partially instructive what you're going to become here. So when I talk about the ectoderm, so ecto means outside, these cells, the part of their genome, the part of their DNA that enables them to become either, epithelial cells or nervous system cells, this part is turned on. Everything else is turned off. So they're partially on the way here. And here, the mesoderm, the part of their genome that enables them to become muscle cells, or skeletal cells, that part's turned on. The nerve part or epithelial part, that part's turned off. And endoderm, these are the genes that turned on to become, the internal organs, some of the digestive glands here. So once they're here, then they can't really switch back and forth here. They've sort of put themselves into general categories here. They're still not specified what part are they going to become. So ectoderm, I can't pick on one of these ectodermal cells and say, are you going to become, a motor neuron? Are you going to become a stretch receptor? I can't do that yet, but I know that it's going to be in that general category. And as the development goes, now more ligands come down, specify what is your fate in the organism going to be. And it's a progressive thing. And so everybody gets their assignment here, you assemble correctly, and now I have a functional, organism here, an offspring here. So here's a closer look here. Here's the ectoderm in blue. It's going to become, skin and nervous system. Mesoderm become muscle, and the endoderm going to become the internal organ here. And now this forms an embryo. And I still, I don't look, all I look like here is these three sheets of tissue here. I'm embedded into the uterine wall here. And what's starting to form here is going to be this group of, this placenta. The placenta is starting to form, because what's going to happen is that these cells, they require a circulatory system. They require, blood to be made here in order to, form this organism. But it may not necessarily be compatible with mom's system here. So I've got a separate system called the placenta, which is going to enable nutrients from mom to go into the developing embryo here. Waste product from the developing embryo to go back into mom's system here. And mom's blood never actually enters the developing embryo. And so that's a good thing, because just genetically, mom's blood type might not be compatible with, the developing, embryo's blood type here. So, that would be bad. That would lead to the death of, both, mom and the offspring here. But no problem. I've separated the blood supply. The blood supplies actually never come in contact with another. And here we are. I've got the developing umbilical cord here. So here's going to be the part where I'm going to plumb this thing into the placenta here. And this is how the exchange is going to go on here. Chorion, just means shell. I've got a protective shell here. And I'm still, isolated myself here from, mom's immune system here, to make sure that mom's immune system doesn't get in here and destroy the, developing embryo or the developing fetus here. So here I can see the placenta is a little bit more diagrammatic here. Here's mom's blood supply, and it makes a pool of blood here in the placenta. And here's baby. So here's the umbilical cord here. The umbilical cord has all these capillary bits that stick right up into the pool of mom's blood here. And now oxygen comes in here, nutrients come in here, waste products from baby, they go out the other way here. And this is going to be how nutrients is exchanged with the developing, fetus in mom here. And it's going to be via this placenta. So blood never mixes here. And, now development goes on here. Here in mammals, we have this structure called the yolk sac. And, we don't have a yolk. So, what's with the yolk sac? And the answer is that if I look in, birds have a yolk sac. And this is the yolk here. So in mammals, we have the same yolk sac because we come from the same, genetic origins here. We have this, but it no longer provides yolk. I don't need the yolk because I got this placenta, which can give me just much, much more energy. I don't have any use for this. But the yolk sac preserves its name because it's still there. And the job of the yolk sac, instead of supplying nutrients, just like it does in, in a bird egg, it supplies the first blood cells. So the first blood cells are manufactured, and this is gonna happen within a couple weeks here. Starts to supply blood cells to the developing embryo here, and this is going to be what gets everything on the road. So fetal heartbeats, they happen 4 to 6 weeks after fertilization. You can hear the pump going. They're moving blood cells manufactured by the yolk sac. They're supplying the nutrients throughout this organism here, because even after you're a few hundred cells population here, simple diffusion now is just too slow. You cannot support that kind of development here, because I can't get it in and out, can't get nutrients in and waste products out fast enough without a circulatory system. So this part comes online really quickly and necessary for survival here. Here's another close-up view of the placenta. So here is mom's circulation coming here, making this pool of blood here, baby's capillary system here, and now bring in nutrients, take out waste products, do the exchange in the placenta, have everything go on their way here. So like we said, this is why mom and baby can have different blood types. I don't need to match the blood type, because the blood never actually mixes. I have this exchange area here to move products in and out of baby here, and this is going to be the umbilical cord here. So this is cord goes to the placenta, goes into baby's belly button here. Belly button goes to the regular circulation, and now baby's on its way developing, and it's got just a very, very large amount of energy, because mom's here continually making contributions to the energy supply, to the nutrient supply. So it's not like the yolk. As soon as that yolk's gone, if you're not done with development, you're sort of out of luck. There is no chance to get any more. With carrying the fetus internally, it means that mom can always eat more food. It can get more nutrients here, keep baby's development going. And for mammals with very complex developmental procedures, because we're complex organisms here, you need that continuous energy supply. So this internal bearing of children, of the young here, this is absolutely essential if you're going to have a complicated organism, because the demands of energy by the developing fetus are so high here. Okay, some terms you got to know. Zygote. Once again, this is the cell that results from the fusion of my haploid sperm and my haploid egg. I make a diploid zygote. As soon as that diploid number is formed, zygote starts to divide by mitosis, making more cells here. Morula, as that cells keeps on dividing and dividing, I get the solid balls of cells produced by the mitotic division of the zygote here, and kind of looks like a berry. This is what the origin of the morula is. It means berry, and I think it's Italian or something. Blastocyst, the morula differentiates. Cells migrate to one end of that solid mass. They make a hollow ball of cells, and this is where the embryonic stem cells pool on one side of the blastocyst here. The blastocyst is going to be what embeds itself into the uterine wall here. Embryonic stem cells is those cells that can become anybody. They're cordoned off in one part of the blastocyst here, and as the blastocysts develop, the embryonic stem cells, they receive their calling. They become endoderm, mesoderm, and ectoderm, and now development is on its way. Embryo, developmental organism until about 8 weeks, and this is just people just let's call it 8 weeks. So there's no absolute demarcation point. This thing is continuously developing. So at 8 weeks, it stops being an embryo and becomes a fetus. And this is just the terminology that the embryology community happens to use to make that call when it's one or the other. Ovulation, the release of the egg from the ovary here. Amnion, the membrane that surrounds the developing fetus. Amniotic fluid, the fluid that surrounds the developing fetus here. So amnion is the membrane, the amniotic fluid. The fetus is floating in this protective fluid here. And, amniotic fluid. Teratogens, these are factors that can cause birth defects. And this is a general term. So this can be anything from like some kind of harmful compound like benzene. It could be a gene that's failed to protect the organ. So teratogen just means something that's going to cause a birth defect here, and some come from the genome of the developing fetus itself. Others come from external sources, some kind of toxin or pathogen in the environment, and result being birth defects here. Let's look at the breast here. So clearly, as soon as the child is born, then in mammals, they need this nutrient milk in order to survive here. And how does that happen? So there are all these cells here that are going to secrete the milk here. LH is going to come down here. I've got other hormones that tell these guys to start making this milk production here. And these are modified sweat glands here. So the modified sweat glands, and they ooze things out through these little pores here at the nipple here. So in a lactating female, you can't see there's no hole here. It's just like you can't see your sweat glands here. You can't see the holes here, but everything's being transported here through these little ducts here, and now provides a very specialized substance of milk to nourish the newborn here. So when you have a newborn, you can't feed them cow's milk. You can't there are supplements that you can give, but they're specially formulated in order to allow the newborn to survive. So it's a completely different diet than as an adult what you're used to consuming here. All right, so here development proceeds. I've got the developing fetus in here, and this is normal position, the head down position. And for birth to occur properly, the head has to be in the down position. Here's going to be our cervix here. And here's the placenta still plumbed into baby's belly button here, giving nutrients, taking away waste products here. And everything is going normally at this point here. And let's look at the placenta. So here's the placenta, and here's the umbilical cord, and it's going to be plugged into baby's belly button here. And on the other side of baby's belly button, here's going to be the umbilical vein here. It goes to the liver here to make sure that, clean up all any toxin that might be making it through here. From there, it enters general circulation here, and then comes back to, back up into the placenta, brings the waste products back here to exchange here. And, what happens is that this is a great system. I can exchange, nutrients and waste products through here. It gets plumbed directly into baby's system here. And when you're born, as soon as you're born, of course, somebody, cuts the umbilical cord here, and that separates you from the placenta. Okay, that's fine. No problem there. But on the other side, this umbilical vein here, there's an enzyme that comes in here and degrades that. So the circulatory system of the newborn, and this happens, within hours here. This part is degrading here, and now I get, a more normal looking, circulatory system here. So, when you look inside of a cadaver, you look on the other side of the belly button, there's nothing there because the enzymes have taken away that tube there because it's no longer necessary. It's no longer, making any, contribution to the survival of the individual here. So it is simply removed, and now the adult circulatory system is basically, configured, and development of the child goes on, and development proceeds here. Here's the position of baby ready to come to term. Development is completed here, and now time for delivery here. So what happens is that the uterine muscles start to contract. As the uterine muscles start to contract, baby's head starts to push on the placenta here. And this is going to start a process of positive feedback. So this is a positive feedback mechanism here, because it's in the interest of the organism here to get this delivery, over and done with as quickly as you can, as quickly and safely as you can, because it's quite a vulnerable time for the mother here. So the uterine contractions start. Baby's head pushes on the cervix here, and there are going to be stretch receptors in the cervix that now send signals up here to the hypothalamus, and the hypothalamus is going to release cause hormones to be released from the pituitary gland, and the principal hormone is going to be oxytocin. So oxytocin is now put into the circulation. Oxytocin comes down here, and it turns out that the uterine muscles have receptors for oxytocin. And when this oxytocin released from mom comes down here and binds to these uterine oxytocin receptors, they contract more. So it's a feedback. I get the pressure here, goes up here, oxytocin, and now more pressure because muscle contract more pressure, and then it goes round and round and round. And as the delivery process proceeds, mom's filling up on oxytocin, and so is baby for that matter, because baby's in that circulatory system. So both mom and baby are filling up on oxytocin. Uterine contractions are getting stronger and stronger, and finally, the baby is going to be able to be pushed through the birth canal here. So this positive mechanism here pushes baby through the birth canal. And remember, the problem here is that in mammals, the brain has gotten so big that the delivery process is complex here. We've reduced the size of the brain as much as possible. You just have the basic instruction set that you need to survive here, but still, it's still a large brain here. And as this contraction goes, it's not like you come straight up. Baby has to be positioned in a very precise way. The head has to be down, and as baby's coming out, the head has to rotate in just the right direction. It rotates in the right direction here, and now baby is delivered here. After the delivery, the hormones are going to cause the placenta to detach. The placenta is delivered after the baby is born here, and baby is now on its way here. So remember, everything happens due to this positive feedback. This positive feedback is run by this oxytocin molecule, causing more contractions of the uterine muscle here. And remember, oxytocin has another function. This is the bonding hormone here. So, once the birth process is complete, mother and baby, they're just chock full of oxytocin. And, when mom sees the baby, you have that very strong bonding, event here. So the bonding event happens, mom bonds with it, baby is going to bond with mom here. And so is the attachment of moms to their children just because of this oxytocin molecule? Well, in large part, the answer is kind of, yeah, this is what makes you have that feeling of bonding here. And that's not a bad thing. It's not, I don't think it detracts in any way from the experience to know what the mechanism is. It just says, okay, I got oxytocin, mom's bonded with the child. It feels great. And okay, I don't have to, agonize on whether it's an automated response or what. It's something that's desirable. It's something that, everybody enjoys. So, okay, let's accept it and move on here. Let's look a little bit about genetics here. So here's the chromosomes in humans here. I got 46 chromosomes. Like we said before, I got big ones and I got small ones. And I can take all the chromosomes in a human. I can take the two biggest ones, I line them, the next two big, and they all form pairs here. They're coming down here. And I've got 23 pairs of chromosomes here. So the two biggest ones I'm going to call number 1, the next biggest number 2, then number 3 here. And I can see that the little markings, the little stripes on these chromosomes, they match because they're homologous, they're the same. One's from mom, one's from dad. They have the same genes at the same location. And so like I have a duplicate copy here. And everything goes fine until I get to the 23rd pair. And the 23rd pair, now this one, these are called the sex chromosomes. And this is what's going to determine your genetic sex here. If you have 2 X chromosomes, then you are a genetic female. If you have an X and a Y chromosome, and the Y chromosome happens to be smaller than the X chromosome, so I can just look at that chromosome and determine whether it's a genetic male or genetic female. If it has an X and a Y chromosome, then what happens is that is going to become a genetic male here. More testosterone receptors. If there are 2 X's, then, more estrogen receptors here. And I can look at all. And so in all organisms, you've got to have the right chromosome number. If you've got the wrong chromosome number, then it turns out that this is going to lead to, very serious developmental defects in the individual here. And so there are, errors here. The most widely known one is Hansen's disease. And that I have, or trisomy 21, Down syndrome. With Down syndrome, I have an extra number 21, and I can survive there because there are not that many genes on it here. So I can survive it. But then I have severe developmental defects here, and that's going to be a problem here. So I can see that that's going to happen, before the child is born here by sampling cells from the amniotic fluid here. I can look at the chromosome count. If it's got an extra number 21 chromosome, then I know it's going to have Hansen's disease here, and it's going to have a certain kind, a syndrome of developmental defects here. We very seldom see a trisomy of the big chromosome because there are just too many genes here. I mean, it does exist, but it's very rare because there's so many genes here that it misrepresents, and these usually have spontaneously miscarried. They do not go to the full term of development here. So I can have multiples or deletions in these chromosomes here, and not so much in the bigger ones here. So let's look at our chromosomes. Here's the chromosome from mom. Here's the chromosome from dad. They have the same genes. One could be dominant, the other could be recessive. They could be matching, it could be the same. So whatever it is, but I've got 2 copies of each gene located in exactly the same place here. And this is going to be important. So this is like the recipe book here, meaning that when they want to read this gene for R, it's got to know what page of that recipe book is on here. They go right to that page, they look for this, they read that gene, they get this. They could read them on, mom's chromosome, they could read them on dad's chromosome. If they're the same, they get the same thing. If they're different, okay, maybe I got 2 different versions of that gene that are now being read, one dominant, one recessive. And so this is how the system is made. But it really relies that this gene has to be in this place. If this gene is placed someplace else, it's just like taking a book out of your recipe book and putting it, in the wrong place in the book. You can't find it because now the mechanism is set to go here, read this gene. If this gene is not there, then the system, can't code that gene, and now that's going to be a serious genetic defect for that individual here. Let's look at another concept here called the genome here. And with the genome, this is the total genetic information carried by an organism. So this is everything. This is, all of the DNA, in, all of the genes. It's all of the non-coding segments of genes. It is absolutely everything makes the genome here. And so when I look at the genome of different, organisms here, so there's a human genome, there's the fish genome, there's the bird genome, and for the most part, it turns out that, there are great areas of overlap here. So most of the enzymes that we use to, to stay alive here, to run the body here, they're present in birds, they're present in fish, because, we've all come from the same, origin. We started out from, being modifications of some, primordial, species here. We've branched out. So we've got strong similarities in terms of genes between all of the genomes here. But there are also differences here. And this is why that, fish have the gene to make gills. We don't, and birds don't. Birds have the gene to make feathers. We don't, and fish don't. And so once a gene has left the population, then you can't put it back there. I can take a gene and I can modify it to work better in, my situation here, whether I'm a human or a fish or a bird here, but I can't sprout a new one. For mammals, let's say, you lived in an environment, and man, it would be such a great idea if you could fly, and you really, really want to fly. You've already given up. You've already lost the gene for feathers. You are never going to get that. There's no facility for, mammals to make feathers because it's just not in your genome. Same with fish here. And let's look at what happens. There are flying mammals, bats, and they cannot, sprout feathers. So they have basically a membrane-covered wing. And so this is their means of becoming airborne because they can fly, but they can't have feathers here. And if you see bats in Arizona all the time, you can see the bats they don't fly like they don't have the grace that you see of birds here. They kind of look like they're about to fall out of the sky there. But what makes bats so successful is that, okay, they might not have the flight characteristics of the feathered birds here, but they have the mammalian brain here, which just is much, much smarter than birds can ever evolve here. So they can, outsmart their prey here. They're just, that is their survival advantage here. I can look at ocean-going mammals, like dolphins or whales here, and they can't sprout gills. That's why they have to come up to breathe. If they don't breathe, they're going to suffocate just like we would with lack of oxygen here. So they have to continue come up here to breathe, to the air here. But again, they have the mammalian brain. So you put a dolphin up against a school of fish here, there is just no contest. These guys can work as a pack. They can collaborate. They corral all these fish there, and then it's just a feeding frenzy of dolphins eating up all these fish that are in this ball here, and the fish, they don't have the brain power to figure out, how do we get out of this? They're sort of, they don't have that mental capacity that mammals have developed, and this is the advantage of the mammalian species here. So kind of remember, if the question comes at you, what's the difference between the genomes between organisms here? That some organisms have genes. Mammals have the genes for a bigger, more calculating brain. Birds have the genes for feathers. We can't get feathers. Birds can't get the superior processing power of the mammalian brain here. And this is just the way that evolution works and how things have worked out in our environment here. Let's finish up our slide here with this last slide here. And this kind of sums up our view of how organisms, including humans, are put together. So let's look at humans here. The human genome makes the brain, the physical plant of the brain, and the mind, your ability to imagine who you are, what you're up to, what you want to achieve in life here. And as individuals, we see this in the news every day. We can choose to cooperate or we can choose to destroy. And it's an open choice for us. There is an element of making a choice here. So, here are a bunch of people, assembling a solar panel here. It's, a homemade, a community project. You can choose to do that. This happens to be a picture of the bombing at the Boston Marathon a few years ago. And, both of these involve, advanced logical thought. People have thought about what they want to do here. And that's what we do. We choose, do we want to cooperate or do we want to destroy? And that's the nature of our species. So, choose wisely and, live your life and go out there and do some good. Okay, that ends our presentation here. Good luck on our exam, and I will see you in the next presentation.